Hi everyone, I'm Duleepa Pathiraja from the Computational and Synthetic Biology Laboratory, Korea University. Today I'm going to present a recently published article for our journal club meeting. And the title of this article is Microfluidics-free single-cell genomics with templated emulsification. This article was published in Nature Biotechnology in March 2023. The researchers involved in this study are mainly from the University of California, Berkeley, and a biotech company called Fluent Biosciences. And this group also consisted of researchers from MIT, University of California, San Francisco, and also from Chan Zuckerberg BioHub. Before we move into the article, let's discuss some basic background information. RNA sequencing is used for the transcriptomics analysis of biological samples, and this is a widely used technique, and I assume that you all know the basics and the workflow of RNA sequencing. Single-cell RNA sequencing, or scRNA-seq, is a NGS-based method that is used to examine the transcriptome of individual cells, which allows to study the cell-to-cell variation in gene expression. And the single-cell RNA sequencing is widely applied in cell type classification, discovery of unique cell states during development and diseases, and for the prediction of cellular regulatory mechanisms. So compared to the bulk RNA sequencing, this method has few advantages, such as transcriptome analysis can be done at single-cell input levels, and the protocol proceeds from the whole cell, so that preserves the integrity of RNA, and high resolution that enables discovery of cellular differences masked by bulk RNA-seq methods. There are two main single-cell RNA sequencing platforms widely being used now. First one is the droplet fluidics platform. One key publication that described this method was published in 2017 in Nature Protocols. The article titled Single-Cell Barcoding and Sequencing Using Droplet Microfluidics. This is based on co-encapsulation of cells, barcodes, enzymes, and lysis reagents into microfluidic droplets. Inside the microfluidics device, single flow of cells are mixed with barcoding hydrogel particles, reverse transcriptase, and lysing reagents. Then all are encapsulated into an oil droplet. Only a single cell is encapsulated into a droplet, so all the mRNA with single cell origin are barcoded with a cell barcode. However, this method requires specific microfluidics device or instrumentation that perform the single cell isolation into droplets. Droplet fluidics platform is now commercialized by 10x Genomics, which introduced the Chromium controller and Nadia instrument from Dolomite Bio, and Tapestry device from Mission Bio. And currently 10x Genomics is the leading in single-cell sequencing methods. The second platform is combinatorial indexing, which add combinatorial barcodes in several rounds of split, pool, and barcoding cycles. So which means first reverse transcription was performed with barcoded reverse transcription primers within intact fixed cells. Then the cells are pooled and split, and then another barcode was ligated, and again cells mixed and split. And this mixing and splitting is repeated for another cycle, and then the cells are lysed. So after several cycles of split, pool, and barcoding, RNA molecules in each cell are uniquely barcoded. So this method does not require physical isolation of cells, but cannot be used with large number of samples. So this method is first published in 2018 in Science, titled as Single Cell Profiling of the Developing Mouse Brain and Spinal Cord with Split Pool Barcoding. So if you are interested in single cell RNA sequencing methods, I highly recommend both those two papers for your study. Combinatorial indexing platform has also been commercialized by Pass Biosciences, and their Evercode whole transcriptome kit is now commercially available. The method described in today's article is more related to microfluidic encapsulation, but does not require a microfluidics device. So that is the main difference. In this method, a method called templated emulsification is used to encapsulate single cells with barcoded templates, lysis reagents and enzymes. Therefore, this method is called particle templated instant partition sequencing, or PIPSIC. Before going to more detail, let's look at a previous study that laid the foundation for this study. In 2018, a study published in ACS Analytical Chemistry demonstrated the compartmentalization of biological reactions in monodispersed droplets. A hydrogel particle is soaked in a reaction mix, for example in a PCR mix. So the molecules smaller than the pore size diffuse into the hydrogel, and then oil and surfactant is added and vortexed. While vortexing, droplets are broken into further smaller droplets, and when a hydrogel particle has been encapsulated, the minimum size of the droplet will be the size of the hydrogel particle. Therefore, the monodispersed hydrogel particles template the formation of monodispersed droplets. So each droplet contains single hydrogel particle co-encapsulated with macromolecules such as DNA and other enzymes and reagents required for the reaction. In the current study, cells are co-encapsulated with polyacrylamide barcoded beads and lysis reagents. First, cells, barcoded template hydrogel particles and lysis reagents are mixed, and then oil was added and vortexed to form the monodispersed droplets. Then the cells are co-encapsulated with the hydrogel particles into oil droplets, and then heat triggered the cell lysis and released the mRNA. So they are captured by poly-DT oligonucleotides on the surface of the polyacrylamide gel particle, and the mRNA are reverse transcribed and the library is amplified with barcodes and then finally sequenced. In PIPSIC, the droplet size controls the droplet volume, and the number of droplets is determined by the container volume. So it is estimated that 3500 cells can be barcoded with 35 microliter barcoded hydrogel templates in a 500 microliter tube, and 225,000 cells can be barcoded with 2 milliliter barcoded hydrogel template in a 15 milliliter conical tube, and 1 million cells can be barcoded with 10 milliliter of barcoded hydrogel templates in a 50 milliliter conical tube. So encapsulation is also possible in 96, 384 or 1536 well plates, scaling the number of samples per study. So compared to the other single cell RNA sequencing methods, PIPSIC can be scaled up to more than 1000 samples and 1 million cells per sample. To prevent the cell lysis before encapsulation, proteinase K, which has minimal activity at 4 degrees of Celsius but which can be activated at 65 degrees of Celsius, is used. And to demonstrate the heat activated cell lysis, cells are stained with casein, which is a fluorescent dye, and showed that before heat activation, dye is localized in the well, but dye is released upon heat activated cell lysis. To further study whether pre-lysis occurs before encapsulation, mouse and human cell lines were mixed, and the barcode rank plot showed typical cliff and knee shape indicating the good separation of cell associated barcodes and the background. And the cell mixing study showed that less than 3% of mouse reads in human transcriptomes, indicating minimal transcriptome mixing and formation of cell doublets. To investigate the feasibility of PIPSIC in cell atlas studies, PIPSIC was applied to healthy breast tissue samples from multiple individuals, and PIPSIC data across participants were combined, and expected cell types were recovered by dimensionality reduction. Then PIPSIC and commercially available 10x Genomics Chromium platform was compared. Cells were clustered in same pattern in both methods. So this shows PIPSIC is comparable to the currently available single cell RNA sequencing methods. Next, marker genes and correlation between gene expression profiles within each cluster was compared, and predicted marker genes are consistent between methods, and gene expression was highly correlated between two methods, PIPSIC and 10x Genomics. Then we will move into a couple of applications of PIPSIC method. Single cell level CRISPR perturbations allow the screening of genotype-phenotype relationships. But in genome-wide scale, it requires millions of cells and thousands of single-guide RNA libraries. Here, transcriptional changes associated with the CRISPR interference allelic series CropSeq library was screened using this PIPSeq method. And CRISPR interference requires the co-expression of catalytically inactive Cas9 protein and a customizable single-guide RNA. This Cas9 single-guide RNA complex binds to DNA elements complementary to the single-guide RNA and cause a steric block that holds the transcription elongation by RNA polymerase that results the repression of the target gene. So in this study, up to 6 single-guide RNA per gene in this library is designed to quantitatively measure the knockdown efficiency across the allelic series. And this library separately expresses a polyadenylated copy of guide RNA, allowing the capture of both mRNA and guide RNA. So CRISPR interference lentiviral library was transduced into K562 cells, and transcription profile and single-guide RNA identity was sequenced with PIPSeq. Single-guide RNA for each gene are ordered from high to low predicted knockdown efficiency obtained in a previous study. So the expected read counts should be in ascending order. The null single-guide RNA is non-targeting single-guide RNA and is used as a control. Using PIPSeq, a gene expression was measured in terms of read counts and compared to the predicted knockdown efficiency across each gene's allelic series. For cells with one guide RNA assignments, previously reported knockdown efficiencies correlated with the normalized read counts. Single-cell RNA sequencing is widely being used for the monitoring of cancer in response to therapy. Transcriptional response of two cancer cell lines, H1975 and PC9, to gefitinib was studied. Gefitinib is an epidermal growth factor receptor tyrosine kinase inhibitor, and H1975 cells are resistant to gefitinib. Shift in transcriptional response was not observed in gefitinib resistant cells, but substantial shift in gene expression was observed in PC9 cells. Differential gene expression also showed increased expression of tumor-associated calcium signal transducer 2 in PC9 cells. In addition, when two cell types are mixed and sequenced, the two cell types can be differentiated solely by their single cell transcriptional profile roughly to the expected proportions. So this is very useful to monitor the progress of treatment and identify the proportion of cells that are resistant to the therapy. Next, PIPSeq was used to study mixed phenotype acute leukemia. Relapse and changes in immunophenotypes upon chemotherapy are monitored using flow cytometry, but using PIPSeq, additional layer of complexity which was undetectable with conventional immunotyping can be detected. Dimensionality reduction identified cell clusters that emerged after chemotherapy. However, these clusters showed similar immunophenotypes in conventional flow cytometry studies. With PIPSeq, a notable transcriptional heterogeneity was observed in these gene clusters, and several new genes that are previously unknown with mixed phenotype acute leukemia were identified with their transcriptional upregulation. In summary, PIPSeq is a fast and simple method of sample processing for single-cell RNA sequencing. Rapid sample processing eliminates the need for fixatives and minimizes the transcriptional perturbations and batch effects. And intrinsic scalability of the method makes it well suited for large-scale cell atlas studies, and this method can be integrated with robotic fluid handling and therefore can be implemented in high-throughput experiments. Finally, this can be adopted for monitoring cancer progression and tailoring new treatment methods. With this, I wind up my presentation for today's journal club meeting. I would like to thank you all for your attention during this presentation.